As Randy Alcorn and Jim Darnall argue in an article for Eternal Perspective Ministries entitled 10 Major Flaws of Evolution Revised, quote, "Mutations are thought to drive evolution, but they cannot increase information. Even mutations which are in some way beneficial, such as antibiotic resistance in bacteria or wingless beetles on windy islands, result from the loss of information. This is the opposite of the vast increase in information required to get from amoeba to man, as proposed in the theory of evolution." End quote. It is true that some of the time, a beneficial mutation does consist of a loss of information. For example, during the DNA replication process, part of a gene could fail to be copied, and this could deactivate a protein responsible for pigment color, with the end result being that the organism now blends in better with its background. These creationists are correct that some of the time, evolution takes place when genetic information is lost. But they're mistaken when they claim that this is the only mechanism by which evolution can take place. On the contrary, there are several ways that mutation and evolution can increase the amount of genetic information. One of these is through the duplication of genes. Due to a mistake in the DNA replication process, sometimes an additional copy will be made of a particular gene, and this change will be passed down to the offspring. Because the organism doesn't presently need two genes that do the same thing, this means that one of the genes is free to mutate and change in a way that could potentially benefit the organism. As we read in Genetic Analysis and Integrated Approach, quote, "In a process called neo-functionalization, a mutation in one of the duplicates could provide a function not performed by the original gene. In rare cases where the new function provides a selective advantage, the gene can be maintained and become fixed in the population." End quote. There are countless examples where precisely this process has occurred and been identified. We're going to look at several examples, but understand that this is just a tiny sampling. As Brandon L. Lodgemon et al. Write in the Journal of Biological Chemistry, quote, "The copper transporter family of integral membrane proteins is ubiquitously found in eukaryotes and mediates the high affinity transport of copper across both the plasma membrane and endomembranes. While mammalian copper transporter 1 functions as a copper transporter for copper acquisition and is essential for embryonic development, a homologous protein, copper transporter 2, has been proposed to function as a low-affinity copper transporter, a lysosomal copper exporter, or a regulator of copper transporter 1 activity, but its function and evolutionary relationship to copper transporter 1 is unclear. Here we report on a biochemical, genetic, and phylogenetic comparison of metazoan transporter 1 and 2, suggesting that transporter 2 arose over 550 million years ago, as a result of a gene duplication event, followed by a loss of copper transport activity." End quote. Nicholas Panchi et al. Provide dozens of further examples in the American Society of Plant Biologists. We're going to read the entire study together. I'm completely kidding, of course. I'm extremely joking. We'll just look at a few examples, although it is a fascinating paper. Quote, "Some examples where neo-functionalization after duplication has likely contributed to duplicate retention include Madsbach's transcription factors involved in the evolution of novel floral structures, 4,5-dioxygenase and cytochrome P450 genes that contribute to pigment variation in caryophyllase, and the recruitment of duplicated primary metabolite genes into specialized metabolite pathways. Some specialized metabolic genes likely arose from neo-functionalized duplicates of primary metabolism genes, and further duplications of specialized metabolic genes have likely contributed to additional, novel biochemical activities. In some cases, these duplicates are implicated in defense against herbivores and microbes, as well as in attracting pollinators. A duplicated NOx transcription factor has acquired a novel regulatory pattern that regulates leaf shape and above-ground architecture in plants. Finally, duplication can contribute to the interaction of plants with other organisms. The duplication of a receptor-like kinase gene originally involved in mycorrhizal symbiosis gave rise to the lysine motif receptor-like kinase, SILYK10, holy fuck, these names are killing me, SILYK10 in tomato, which likely adopted a new role in nodulation with clear adaptive significance." End quote. I'm on Patreon. There's a link in the description if you'd like to support my content. I would really appreciate it. There's also social media links in there if you want to follow me there. I'm trying to build up some of those followings. I would really appreciate it. Make sure to subscribe for more videos if you haven't already. Hit the bell, hit the like button, post a fucking comments. All that fun stuff that's standard on YouTube. You know the fucking drill. Thanks for watching. Hope you enjoyed this.